Thank you, Sarah, and thanks to the organizers for inviting MaxCyte to come and talk. So MaxCyte is a delivery platform company. We use this delivery platform technology in areas of small molecule drug discovery, protein production, cell and gene therapies. Specifically in cell and gene therapies, we license our platform to other commercial developers and are also working on developing proprietary therapeutics that are uniquely enabled by this delivery platform. The way the platform works is we can take any cell, any molecule, and load up cells with different molecules. Can you all hear me? Okay, perfect. And we use the MaxCyte GT instrumentation platform, which is a closed system. It's an automated platform. It is GMP and regulatory compliant. Essentially, what we're trying to do is we're trying to pre-program a cell to engineer the biology of a cell such that the engineered cell has more desired biological characteristics. And the way we do this is by doing pharmaceutical engineering inside the cell, by loading a cell up with molecules to control the molecular flux along different pathways so we get more effective biological outcomes. The cell, when used as a tool in drug discovery research, becomes a screening tool. You can take over the cellular production machinery and use it to make therapeutic antibodies or vectors or vaccines. It's a biomanufacturing application. Or when the cell is given back to the patient, it becomes a therapeutic application. So the salient features from a performance perspective, the first order of doing any modification is do no harm. So we want to make sure it's non-toxic, doesn't damage the cells. The basic physiology and function of the cells is maintained. It gives us high viability and loading efficiencies. Scalability is a feature designed into the performance of the system where we can run using different families of single-use disposables, anywhere from a few 100,000 cells to a few 100 billion cells. And it takes us about 20 minutes to run 200 billion cells through the system. So it's an automated, rapid, large-scale, commercial-sized system for delivery of molecules into cells. I already talked about the quality and the regulatory aspects. We have a CE mark. We have an FDA master file. The system is being used in over a dozen clinical trials in multiple countries. We currently support also one commercially marketed oncology therapy product, which is on the market in Japan. So specifically, I'm not going to talk about the discovery research part, but talk about the cell therapy part and what we're doing in developing novel therapeutics enabled by this platform. So starting off with just stem cells, to give you an example of the use of stem cells, using some surrogate molecules that are loaded into stem cells, a variety of stem cells, hematopoietic stem cells, mesenchymal stem cells. Essentially, the sole purpose of this slide is to show you we can get very good viability and more than 90% efficiency in terms of loading stem cells. Not only do we get the high efficiency, we can control how much loading there is per cell. If you look at the distribution itself, it's very uniform and consistent. So why would one want to modify cells? And to some extent, I'm going to borrow an example from mesenchymal cells. And a lot of companies are working with mesenchymal cells where you culture the cells a particular way and use those same cells for many different applications. But the cell doesn't really know what biology it needs to manifest. It doesn't understand whether it's being delivered back into the brain, into the heart, or into the bone. So here's an example where we brought an orthogonal mechanism of action where we can use the same mesenchymal cell in addition to its paracrine secretion activities to become a localized in vivo bioreactor to pump out BMP-2. BMP-2 is a validated pharmacological agent for bone regeneration. And combining two orthogonal pharmacological mechanisms of action, what we see is improved time to regeneration of bone and better quality in terms of the tensile strength in animal models. So again, shows you the power of actually being able to engineer a cell to combine multiple modalities of how effectively you can enhance the potency of a cell therapy product. Beyond just adult stem cells, we were also working in the field of using our platform as a DNA fingerprint-free method to generate iPS cells and to convert iPS cells into lineage-committed precursor cells for various therapeutic indications. Here's an example of a grant we have with a collaborator at Johns Hopkins University that's funded by the Maryland State Stem Cell Consortium in driving towards generation red blood cells from iPS cells as a potential therapeutic use. Those two examples I shared with you were transient modulation using molecules at the cytosolic level. But we're not just restricted to that. We can also use a platform as a delivery platform for delivering gene editing tools into cells. Here's an example from a presentation given by one of our partners, Sangamo, last year, where they basically used our platform to deliver zinc finger nucleases into a variety of cells. This example is with hematopoietic stem cells, where what they show is they get 30 to 50% functional gene correction in hematopoietic stem cells. There is no long-term safety or toxicity issues in animal models. There is engraftment in non-skid mice, and long-term phenotype of the corrected gene is being maintained in the animal models. Some of these programs, Sangamo has advanced into IND filings and into clinical studies now. By the same token, another example in terms of gene editing tools, this is with the CRISPR-Cas9 system. We can efficiently deliver CRISPR-Cas9 in multiple formats. The example shown here, the top panel shows knockdown using plasmid DNA, the bottom panel, the left panel at the bottom shows knockdown using messenger RNA encoding CRISPR-Cas9, and the right panel shows not just the knockdown, but coming in at the specific site of knockdown to insert a correction sequence. And we're getting about 50% efficiencies of knockdown and about 20-25% efficiency of correcting in the entire cell population. So this really enables companies with such gene editing tools to start thinking about how you can use a plug-and-play approach to go into the clinic in a very reliable and reproducible manner. So essentially, how we use our technology, we're trying to solve two of the biggest problems in developing cell therapies. We want to make cell therapies more effective. So we're trying to reduce the biological risk of developing cell therapies by customizing the biology so that we can enhance the potency and hence get to long-term durable clinical responses. The second problem we're trying to solve in developing cell therapies is to take away the manufacturing scale-up complexity and cost issues associated with large-scale clinical commercial delivery of cell therapy products. And it's really a plug-and-play platform because you can basically substitute out whatever molecule or whatever cell, and essentially the process is identical. We have, as I said, a master file with the FDA, and we've supported multiple IND applications by authorizing cross-referencing of this master file. So in addition to licensing the platform technology, we ourselves are involved in developing certain niche therapeutic opportunities that we're very excited about. One such therapeutic opportunity is in the area of autologous dendritic cell vaccines as active cellular immunotherapy. This is commercially marketed by our marketing partner in Japan called Medinet. And essentially the basis for developing this product was demonstration that compared to the gold standard, we were getting a log order magnitude increase in antigen-specific CD8 positive T cells in a Th1 polarized manner compared to the gold standard dendritic cell vaccine that is being used. So essentially, again, focusing on enhancing the potency and asking if you enhance the potency based on the hypothesized biological mechanism of action, would that lead to durable clinical responses going forward? Now, this, beyond Japan, has also been licensed into a couple other territories in the Asia-Pacific region. We're actively seeking partners in other geographies specific to this particular product. The other product I'm going to spend maybe the next 5 minutes on is in the area of chimeric antigen receptor modified immunotherapies. So really our value proposition in this particular product development experience is we want to develop a commercializable method for manufacturing that takes away a lot of the cost complexity and delivers an effective means of delivering CAR molecules into cells so that we can skew the therapeutic index, have the same anti-tumor activity, but not have long-term toxicities associated with gene therapy delivered CAR molecules. So this whole field has seen a lot of activity in the last 2 years or so. Majority of this has been driven by retrovirus or lentivirus encoded CAR molecules targeting the CD19 antigen. And what has been shown is the clinical response has been just phenomenal, especially in ALL where people are reporting 88 to 90% complete responses in patients who have been treated. The challenge, however, is we have 30 years plus of safety data where you can wipe out normal B cells, which also express the CD19 antigen, and still effectively clinically manage the patients by IVIG infusions. But when you think beyond CD19, there's no other antigen where you have that level of safety. And when people have tried to expand the use of viral vectors into solid tumors, they've had significant adverse events, including patient deaths. So this whole field, specifically just focused on CD19, has seen over $1 billion of investment in the last 12 to 18 months. Okay. So the whole process of using viral vectors is very complicated. It takes about 2 weeks. You have to make the virus. You have to transduce the cells. You have to control all the quality aspects of all the raw materials. You have to control your virus. and you have to control the transduced cell product at the end of the day. So there are multiple challenges from a manufacturing perspective. It is really a gene therapy product, so there's long-term patient follow-up which is needed. There are significant issues, as I described, of on-target, off-tumor toxicity, because there is no uniquely restricted antigen which is only expressed on tumor cells. And a lot of antigens which are sources of antigenic targets for monoclonal antibodies are also expressed on normal tissue, albeit at a differential level. And the challenge is, how do you now control on-target but off-tumor toxicity from a long-term expression? So people are thinking about using suicide genes and other types of approaches, but the challenge really is, how do you actually implement some of these suicide gene therapies when this is a commercially viable therapeutic approach going forward? And can you really expand beyond CD19 into solid tumors? And of course, the commercial implications in terms of manufacturing, scalability, cost of goods. There have been multiple sessions at this conference that have addressed that issue. So our approach is to address each of these challenges. So we're using messenger RNA encoding chimeric antigen receptors as a substitute for viral vectors. We believe this gives us an opportunity to develop a safer, non-viral alternative approach and allows us to expand beyond CD19 into solid tumors, giving us the ability to skew the therapeutic index, transforming CAR immunotherapy into a commercializable product. We currently are in 5 clinical trials with academic collaborators using mRNA CAR loading against different antigens in different indications in expanded T cells and expanded NK cells. And we have gone one step beyond that, where we have issued IP in the context of loading mRNA CAR into freshly isolated cells without requiring any expansion, to try and bring CAR immunotherapy away from the centralized manufacturing paradigm into a transfusion medicine, blood banking type of a manufacturing and delivery paradigm. So I'm going to share some initial data. So the first-in-man studies were initiated in collaboration with the University of Pennsylvania, Carl June's group, a couple years ago, using mesothelin as the target antigen, because mesothelin is also expressed on a variety of pleural and pulmonary tissue. The challenge was really the hypothesis that using mRNA CAR will allow us to skew the long-term toxicity profile. There was a publication which came out in press in February of this year that summarized the clinical response from the first cohort of 4 patients treated. Essentially, what was reported in there was, for all the patients, they were effectively able to manufacture the product. The product had more than 95% viability, consisted of CD3 T cells, and more than 95% of the cells expressed the CAR molecule. The clinical observations to date have been that we haven't seen any toxicity to mesothelin, which is really the primary objective of that particular study, given the widespread and diverse expression of mesothelin. We're seeing persistence of anti-mesothelin CAR T cells, the ability of these CAR T cells to migrate to sites of primary and secondary metastasis, and we're also seeing anti-tumor activity. What is really very exciting and encouraging is, in addition to seeing all of these responses, we've also seen in 2 patients in this early study measurable reduction in tumor burden based on imaging studies. And we're seeing in all the patients treated in this reported study the ability to see epitope spreading, where novel immune responses are being generated against tumor antigens to which there was no pre-existing immune response prior to treatment. Now, this has not been reported in any of the lentiviral vector studies. So this is kind of really exciting and warrants further development. So what we believe this gives us is the ability to say that mRNA CAR looks like a viable alternative for lentivirus or retroviruses. And the next step we're asking is, do we really need to expand cells or not? And our concept is to take a leukopheresis product, take the T cell fraction or the lymphocyte fraction, have mRNA CAR be available as an off-the-shelf therapeutic, loaded with the MaxCyte delivery platform, wash the cells. In a single manufacturing run, we believe we can make about 8 doses for infusion, aliquot them and cryopreserve them for multiple subsequent administrations into patients. So we can reduce the manufacturing time and complexity from 2-week process to something that can be done utilizing closed manufacturing systems, automatable platforms, single-use disposables that can be done at point of care in a matter of a few hours. And so essentially we have in vitro and in vivo data suggesting that this is feasible. We have issued patents describing therapeutic composition matter as well as methods of manufacturing thereof. And we are on track to get into IND studies next year using this transfusion medicine strategy for mRNA CAR. Okay? In that context, we're actively seeking strategic partners for the therapeutic programs, and we're always delighted to talk to anybody who's interested in utilizing our technology and how we can enable improved potency and manufacturability for your cell therapies. So thank you very much.